we've heard some colorful research already. We're going to hear some more about colors. But first, we're going to go back in time a bit. Well, more than a bit. We're going to go back some 150 million years. And the world was a lot less colorful back then, because according to the fossil record, 150 million years ago, that's when probably the first flowers evolved. And that flower presumably looked a little bit like the extant magnolias, that is a hurled flower, white, fused carpels. And then following that first angiosperms, we see the origin of other plant lineages, such as the water lilies, the eudicots, and the dicots. And then towards the end of the Cretaceous, and that is roughly 100 million years ago, we see that there's an enormous radiation in the flowering plants. And the diversity of flowering plants that we see today really stems from that moment in time, that there's a burst in species, and that burst in radiation in flowering plants that coincided with an enormous radiation in animals, particularly insects, that visit these plants. And plants are rooted in the ground, so they need a vector to transport their gametes, their pollen, and many plants use insect as those vectors, as pollinators. And in turn, insects, they get usually a nutritious reward from these flowers, nectar or pollen, so they also benefit from that mutualism. And in his writings to a colleague, Darwin was already bewildered by that enormous radiation in two organismal groups that are in principle not related and that happen in a comparatively short time. And he referred to that as an abominable mystery. Now, the research in my group is about the coevolution of floral optical properties and pollinator visual systems. So how have flower color and color vision of pollinators co-evolved or not, and how has that created the enormous diversity of flowering plants and insects that we see today. And in our research, we stumbled across a number of species that are not just colorful. Most flowers, they're fairly matte. They have a color that is similar regardless of the angle of observation, but we found that numerous plant groups, they have glossy flowers. And here on the left you see a buttercup, a famous European plant group that has these yellow petals, and in addition it has this glossy appearance. It's as if there's a layer of butter on it, hence the name. And we see this glossy, mirror-like reflection has evolved in numerous plants, and the signaling structures on the flowers, such as the red bean, that's a bird-pollinated bean flower from Australia. In the bottom right you see a Dimorphotheca, that is from South Africa. And indeed, if we plot these glossy flowers, we see that they're rare, yet they are widespread across the phylogenetic tree of angiosperms. And by the way, yesterday we heard a nice talk by Vincent de Bar about the iridescent butterflies. These glossiness is not the same as iridescence. It's primarily a change in brightness. So it's this now you see it, now you don't, bright, shiny appearance. There can be some iridescence involved, but it doesn't have to be. And as this glossiness is quite widespread across angiosperms, we see that indeed it's quite common, widespread phylogenetically across the tree of life, such as the brilliant Morpho butterflies. They're iridescent, but usually they also have a flashy visual appearance. And other insects group, jewel beetles, of course, they have these brilliant shiny colors. And vertebrates, fish, amphibians, etc. And then we wondered, what does this shiny visual appearance mean, and how does it contribute to the visibility? And with we, I mean our collaborators on the HFSP, that is Michio Kinoshita from Japan. She is here at the conference and she has a poster. And Daryl Kemp from Macquarie University, Sydney. And we predicted that there are several peculiar things with this flashy visual appearance. For example, the flash effect that these glossy flowers and glossy animals create, it's quite likely that it overwhelms a visual system. Because, for example, there's the shoal of fish. If they're glossy, then if they move all together, then suddenly you get this bright flash of light. And we see similar effects with flowers and butterflies. And then the flash has no specific color. It's just very bright. And then finally, the dynamicity, the changeable nature of the shiny, glossy visual appearance that is quite paradoxical, because signal theory predicts that if you use signals to be recognizable, they should be memorable. And if signals should be memorable, they should be consistent in time and space. But these changeable, dynamic visual effects, they're very changeable in time and space. So we predicted that this flashiness, glossiness, shininess, it comes with a behavioral trade-off in visibility. On long distances, it's probably helpful to be glossy, because it creates a long-distance visual cue that is similar to the flashing lights of an emergency vehicle in your rear-view mirror, or a lighthouse at night, very conspicuous. But then the lighthouse at night, it draws your attention. It's very conspicuous, but it's not the kind of light you would use to read a book in. It's not good for short distance readability, so it undermines the discriminability. And here on the right you see the cover of my thesis. It's glossy and 10 years old almost. And you see the right half, it's slightly bent in the mirroring angle to the window, and then you see the right half which is in the mirroring angle to the window. It's more difficult to see the text and the pictures because of the glossy layer. So we tested exactly that with bumblebees. We tested what does gloss do to visibility. And that was at the University of Würzburg with my colleague, friend Johannes Spese. We tested how do bumblebees respond to artificial flowers that are identical in everything except the degree of glossiness. And here you see some examples of the artificial flowers. They're epoxy replicas of real flowers, so they have real flower surfaces at the micrometer level facsimiles, and they're either glossy or matte. And because they're epoxy replicas, we can add pigment to it, and we can test the combination of pigments and structure altogether. Now, what is a matte flower? So here you see two examples of very matte flowers, purplish flowers, and in the second column you see a cross-section. These matte flowers, they have clear cone-shaped epidermal cells and interior structure and a purplish pigment that creates the overall color. Now here you see the reflectance spectra. They're purplish because there's a peak in the blue and a reflectance in the red. Now this angle dependence, that's not so easy to measure, but one way to do that is to illuminate perpendicularly and then measure the reflectance under different angles of detection. Now if we plot the wavelength, the reflectance at a given wavelength at a function of observation angle, we get the black dots that we see here. So here you see the black dots, and we illuminate perpendicularly, and then we measure at a given wavelength under different angles. And we see that those dots neatly follow the red curve, which is a cosine fit. And that means that the reflectance as a function of detection angle follows a cosine. That means it's a perfect diffuser. In physics, that's known as a Lambertian diffuser. It's just perfectly diffuse. Now, if we plot that in 2D, we create a heat map. At the perpendicular illumination angle, we see the reflection pattern that we saw in this graph. And then if we expand that we also change the angle of illumination, then we get in the heat map that looks a bit like a mountain landscape. Comparing that to, so as I said, that means that the cone-shaped epidermal cells that we see that are most common in flowers, they act as diffusers. They create a perfectly matte visual signal. Now, how does that compare to glossy flowers? Glossy flowers have a very flat epidermal surface that reflects light like a mirror, hence the glossy appearance. And there can be some pigment underneath, which creates, for example, in the case of the buttercup, a yellow overall pattern and this specular reflection. Here are the reflectance spectra. Then if we plot the reflectance as a function of observation angle, we see that it clearly deviates from the cosine. So it's not diffuse at all. And in the heat map, that looks like this. So the reflectance is higher at the mirroring angle, which makes sense. That's glossy reflection. Then we created our artificial stimuli in different pigments, different reflectance spectra, but then within a type of pigment, they're either glossy or matte. And we see the gray triangles here. They are the gray triangles. They clearly deviate from the cosine, particularly close to no normal. So these are very glossy or matte artificial stimuli. Then we wanted to test the impact on visibility. And we did that in an arena. Here's the top view of the arena that we used to test it. So it's a top view. So here at the bottom you see the light source, and right below the light source is the entry hall where the bees enter. And when a bee enters the arena, it sees roughly this. It sees the wheel with 6 stimuli, 3 of which are glossy and 3 of matte, and in different combinations. So we first tested, do they care, the bees, if a flower is glossy or not? Then we started with dark blue stimuli, and it turns out most of the bees innately prefer the stimulus that is matte. So this is significant. So we let them make 10 choices, and we see that most of them prefer the matte over the glossy one. But not if we give them yellow stimuli. They're much more indifferent. They don't really care. Next, we wanted to know, so that they don't care so much when it's yellow, and that's probably because if it's yellow, you see that the gloss is much less prominent than on a dark background, because surface reflections are just more conspicuous on a dark background because there's less reflection coming from the interior. So on a bright stimulus, the gloss doesn't add much. Next, we wanted to know, can we train them to associate the degree of surface gloss with the reward? So they have an innate preference, and we train them to the opposite. And then they can. So this graph should show the original data and the model fit, but somehow it only projects the model fit, so you have to believe me, the original data is there. Anyhow, they can learn to associate just the degree of surface gloss with the reward, regardless of whether the stimulus is blue or yellow. Next, we wanted to know, what does the gloss do to color discrimination? That's the cover of my thesis. Does it undermine discriminability? So we created color pairs that are easy to discriminate, which is what you see here. These are color pairs, stimuli, color pairs that have a quite high color contrast. Blue and green blue, yellow and orange, and the bees don't care. They can always discriminate the two colors regardless of whether the set of colors is glossy or matte. But if we make the color discrimination task more difficult, we give them 2 shades of yellow or 2 shades of blue that is quite similar. We see that the glossy stimuli are significantly harder to discriminate than the matte stimuli of the same color pair. So glossiness undermines fine color discrimination in bees. And this probably explains why the majority of flowers has surface structures that create a matte visual appearance. These cone-like epidermal structures, they occur in about 90% of angiosperm species. And presumably this makes sense more broadly in that having a diffuse, a spatially consistent visual signal helps your visibility. And conversely, creating a flashy visual effect might confuse, might make it more difficult for, say, a predator to locate you. But next we wanted to know, what is the long distance effect of this shiny visual appearance? So we did that in a Y maze. We gave the bee a stimulus. We trained the bee to associate the stimulus with a sugar reward, and then we moved the stimulus back in the Y maze, and either the left or the right. So it knew that it had to find the stimulus, and then it didn't know where it was, and then sometimes it was glossy, sometimes it was matte. And in the other arm of the Y maze, there was nothing. And then distance is the product of visual, so visual size is the product of the size of the object and the actual distance, and we express that in subtended visual angle. So for example, the first row from my point of view has a larger visual angle than the last row. So It's a product of the size and the distance to the object. And then when the bees are close, they don't care much. They see the glossy and met stimuli similarly easily. But then at more long-range detection tasks, and again this data point should have shown the original, this plot should have shown the original data and the fit, but well, it looks better without the noisy data, I must say. But anyway, at long distances, it turned out that the glossy stimuli are easier to discriminate than the matte ones. So there's a trade-off in terms of there's an increase in detection at long range, but it undermines discriminability short range. Now lastly, other things we're doing with the consortium is we're testing similar predictions with flashy butterflies. In Daryl Kemp's group, they've just published some work on it with flashy butterflies, sulfurs and hyperlimnus, and how the flashiness, the changeability determines the discriminability and the attraction of partners. And PhD student in my group, Tom Veldhuis, together with Barbara Gravendale from Leiden and Naturalis, we're testing how shiny orchids that are pollinated through sexual deception, that is, these orchids, they mimic a female insect that is ready to mate. And they're pollinated by males looking for the mate. And they use shiny visual effects. So we're studying that as well. So to summarize, shiny visual effects are overall rare but widespread. They confer visual trade-off, and this has implications of fundamental and applied relevance. And lastly, I want to thank Human Frontiers. I got the faculty position I have now thanks to the grant. It helped me negotiate a position. I would not have had a job otherwise, probably. And of course, my collaborators. Thank you very much.